thank you very much for this opportunity to be with you again. I hope now you have your saliva flowing better. So we're gonna change gears a little bit. We're gonna change the subject, and we're gonna talk a little bit more about science, or more like principles of caries, more than the intervention that we were discussing with the kids before. So again, so the sugars is the main agent and the enemy, because it produces acids from the dental biofilm, which in turn create demineralization and lesion formation. And there's a direct and undisputed direct relationship. And I think this is simple, but it has to be emphasized, because most of the time, sometimes we get a little bit lost when we talk about caries, and we confuse people saying, this is a multifactorial disease, so everything is important. I think we need to be clear and show the people, the patients, that the cause of the problem is their consumption of sugars. Of course, the toothbrushing is important, but the main thing is the diet. And we need to be clear about it, because if you want to operate changes in people and what they do, you need to make sure that the message is simple and clear. So we already know that free sugars cause the disease, but the role of the other nutrients in the diet is less clear. This is where we started our research line. So everyone in dentistry talked about sugars. And the idea is reduce sugar consumption, all right? And what do I put instead? I still want to consume 2,000 calories per day, but what should I eat if I'm not eating sugars? If I delete sugars from my life, what can I put in? And also, our hypothesis was that dietary proteins and unsaturated fatty acids may counteract the deleterious effect of sugars on the biofilm and the hard dental tissues. We had the suspicion that fatty acids and proteins could be beneficial instead of something harmful. The typical way to see caries in terms of diet and nutrition is that sugars is the enemy. But we don't know what happens with other nutrients or molecules that are all the time in our diet. Nobody, as far as I know, eats only sugars as the base of their dietary pattern. They eat different things, right? We all eat different things, but the role of the other molecules involved is unknown. And so our main aim was to determine the effect of the other nutrients on the caries process. After several years, and we're still doing that, we concluded that unsaturated fatty acids present, for example, in oils in general, olive oil in particular has some benefits, but in general, oils, nuts, olives, etc., they have unsaturated fatty acids that are beneficial for cardiovascular disease. But we've seen in our experiments that when you consume that, there is a neutralization of the effect of sugars on the biofilm. Now you would say, well, I know that fatty acids or lipids are non-cardiogenic, right? So this is not our point. Our point is that when you consume sugars, and also you have a lot of beneficial oils, fats in your diet, then you can neutralize the effect of the sugars. And that's what we've shown in multiple experiments. Also, the egg ovalbumin is beneficial. Proteins in general tend to buffer the effect of acids, and in that term are beneficial. And also protein can protect the enamel against the demineralization. So it's a good thing to have protein consumption. And finally, in our research, we've shown that antioxidants and polyphenols that are contained in many foods, but mainly in berries and red fruits, fruits and vegetables in general, they are also beneficial for counteracting the effect of the sugar. So they counteract the effect of sugars when they are present together with these other nutrients. So we published this, and the idea is that from a symbiotic biofilm, we go to a dysbiotic biofilm when we add sucrose or starches or the addition of sucrose and starches. But if you add beneficial nutrients and decrease a little bit sugar consumption, then you can restore the symbiosis in the biofilm and come back to a healthy state. This is what we did over the years. Now, well, this is something that I wanted to show you. This is Jocha and Christina. They were exchange students that were in Talca, in my city, processing and analyzing some of the data that we collected, and they showed that this is the caries experience. So when you eat carbohydrates, there's a direct relationship. So the more carbohydrates you consume, the more caries you have. And same thing, but the opposite was with the proteins. So the more protein you eat, the less caries experience. And this is her thesis, and now has been submitted for a publication, and we hope it'll be available in the journal Nutrients soon. Right, Katerina? Yeah. She was the co-advisor, so. Again, we already said that. I say to my students, I don't want you to memorize anything of what I'm saying, just 4 things. 4 things. For caries prevention and caries management, which is not the same, caries management, you need to learn 4 things. And again, dietary counseling and monitoring first, because this is the cause of the problem. Biofilm control, oral hygiene, but not eliminating the biofilm, which is something that we used to say in the 90s and the early 2000s. We eliminate the biofilm. We eradicate the Streptococcus mutans. That's what we used to say. We can't really eliminate the biofilm. What we do is we disrupt the biofilm. We remove it periodically so it can be reorganized and it becomes healthier. And this is the principle behind oral hygiene. We are not cleaning perfectly and eliminating all the biofilm. It's impossible. I mean, the biofilm is, if you saw the bacterial cells, you would say, well, yeah, really, it's impossible. It's everywhere. But what you want to is to have lower amounts of biofilm. About fluoride, I'm not going to talk because Core spoke about it, and you all know too much about fluoride. And what this lecture is about is the 4th leg of this table of prevention, and it's salivary function, the forgotten component, I would say. So saliva can be a predictor of caries risk. And it's been used along with other factors. So when you see the karyogram, for example, are you familiar with karyogram? It's a software that allows you to assess caries risk. You know karyogram? Well, there are others like Cambra or any system you want. They tend to use saliva. And so it would be, the idea behind caries risk assessment is that you have different factors that put together will tell you whether the person will develop new lesions or not, right? This is the principle behind caries risk assessment. The problem is that this is playing magician, and you need to guess what's going to happen. And among all the models and looking at the evidence, there's no sufficient evidence available to affirm that any method is effective in caries assessment and prevention. So we don't have a tool that is perfect in terms of estimating the risk of someone. So we need to keep doing research, and this is why we are using saliva to do that. We are looking for certain biomarkers of risk using saliva. This is what I want to present to you. So saliva is originated from major and minor salivary glands. 99.5% of saliva is water. So you would say, well, this is a non-important fluid, but it's really important. That 0.5% of the saliva is like a tissue. It has lots of things. Among the many components, saliva has calcium and phosphate that are key in the mineralization and remineralization processes. It's been reported that increased salivary pH and adequate buffering capacity has been associated with a lower incidence of dental caries. But that's it. I mean, this is the only thing that we say, well, we all say. The thing is, the evidence for that is weak and is questionable. So we need to keep doing research. So saliva is important for lubrication, moisturizing, digestion, buffering, remineralization, antimicrobial properties, and taste perceptions. Actually, when you have your dry mouth, right, because you're running or everything, you can't perceive the taste of the food, and that's something that happens with older adults when they lose salivary flow. They start eating a lot of salt and sugar, and they get worse in terms of oral health because this is the only taste they can perceive. This is important, the lubrication of saliva. I already told you that saliva is the most important anticaries agent. We published this paper showing how this is an in vitro model. And so in our in vitro model, we put bacteria, enamel, and culture media. Culture media is like food for the bacteria. If you put saliva, 0% saliva and 10% sucrose, in other words, pure culture media, you have this kind of demineralization in enamel, in green, and in purple, in dentin. If you put to the media 5% saliva, and you still keep the 10% sucrose, look at the drop in demineralization that 5% saliva in the media creates. It's amazing. That's why saliva is so important. And if you go down to, increasing the amount of saliva, you see that you practically neutralize everything. So high quantities of saliva can almost completely inhibit sucrose-induced karyogenicity, while even small amounts exhibit a dose-dependent caries protective effect. This is what I'm talking about. I'm a prosthodontist, oral rehabilitator. How do you say this here? No? Prosthodontist? I'm a prosthodontist, so I see the difficult cases with people that are, we call them the heavy metal generation because they come with a lot of metal in the mouth. And the problem with that is when they are fine up until 65, 70 or so, and all of a sudden they start a process. It's called rapid oral deterioration. And they start creating new caries lesions in places they didn't have, and they start having a lot of oral health complaints. And that situation is critical, and the reason is because they have a decreased salivary function. And for people like those, you would observe caries lesions appearing in unusual places, difficult to treat. So what do you do with that? Extraction. There's no other possibility in that kind of lesion, right? So to start doing salivary research, we needed to know when to collect the samples of saliva, because our theory was that this was a circadian, not our theory, the evidence showed that it was a circadian rhythm of secretion of different things. So we wanted to study the circadian fluctuations. So we took 8 healthy young people, of course, dental students. This is what you do when you want to do an experiment. And we collected saliva at 7:11, 3 p.m., 7:11, and 3 a.m. Yeah, I know, I'm cruel. We paid for that. We paid for that. Yeah, I had a grant. I had a grant, and the grant had the money to compensate this. But we wanted to know what's going on during the night, and we measured pH, calcium, phosphate, nitrate, a lot of different things. And we showed that salivary molecules have circadian variations within the person and inter people. So our, for example, lactic acid has this curve. Nitrate and nitrate has that other curve. And ammonium, for example, peaks at night or early morning. And this is interesting for us because somehow we wanted to know how to do the experiment, but at the same time, kind of you understand certain things. For example, that overnight, in the morning, you have more lactic acid, and probably the toothbrushing early morning is important because you have more acid in your mouth. Or the nitrate, which is beneficial for oral health, peaks after lunch because nitrate is coming from green leaves. So this is something that we learned, and it was important too. So in the literature, we have like 6 different approaches, or there are 6 different approaches that we can take to detect biomarkers. 1, salivary pH. 2, salivary flow rate. You're familiar with that, so I don't want to go into details. The salivary buffering capacity, the salivary microbial composition in terms of S mutants or mutant streptococci, lactobacilli, or microbiome profiling. All of those are being tested regularly, and you would find research, old research, and new research looking at all these kind of things, especially microbiome, the microbiota, the oral microbiome is being analyzed at a very specific level. So now we have a whole overview of all the microorganisms, and there are more than 1,000 different species, and we can practically identify them all with the techniques that we have now in place. But this is less explored, and this is the ones I want to show you because this is what we did. The biochemical composition of saliva in terms of calcium, phosphate, and others, and the salivary level of specific proteins, including IgA, antimicrobial peptides, proteomics, or lipidomics. So let me show you briefly. This is the work of my PhD student that was here in ACTA for 6 months doing experiments. And so she worked under this grant doing these experiments. So we recruited 40 people, 20 without caries or caries-free, and 20 with high level of caries lesions. They had at least 8 non-cavitated lesions or at least 4 cavitated lesions, big ones. And so we did the salivary collection. We measure with reflectometry. This is the apparatus we use. We measure the antimicrobial peptides, the proteomics, and the lipidomics. Let me show you the results. So this is the device. It's from Merck. It's a device that allows you to measure in a scale all these compounds. So in terms of calcium has an inverse relationship to phosphate, and it was difficult for us to understand. So what happens, why calcium and phosphate levels are different in saliva? And in caries-free, calcium was higher, And that means that in a stable environments, there's a remineralization. And that's why the caries-free people, had this trend to remineralize. And there's a continuous mineral deposition into enamel. Whereas in the high caries people, we observe higher phosphate. And the reason is because the frequent acid attacks to the enamel release more phosphate. Phosphate is easily released from the enamel. And that's why we detected more phosphate in the saliva of people with high caries risk. So this could be an interesting biomarker for the future. I'm not saying or not arguing that any of the things that I'm going to show you here are ready to use. It's just, learning. At some point, someone is going to take interest on this and is going to develop a device and they're going to sell it. And I'll be millionaire, I hope. No, that's not my interest. But and so there's a critical, systematic review on the potential proteins that are related with dental caries. And there are many. The problem with this systematic review is that some of the proteins that have been tested, for example, IgA is the one that is secreted by the maternal milk, the breast milk, and gives a lot of protection, immune protection, right? And it's interesting because that IgA is, in our study, was very much related with health. But there are some studies being reported that show that IgA is negatively related with caries. So that's why I say, we still lack a lot of information. Maybe one of the reasons is because you take saliva in different moments of the day. And as I showed before, there's a circadian variation. So the technique and the method matters. So this is our study that we published some years ago about the IgA. You see, high caries people have low levels of IgA. Caries-free people have very high levels of IgA. This was done by Western blot techniques. It's a different technique. It doesn't really matter. But it's a detection of the actual protein. Then we move to different techniques. These are much more complicated. Are you familiar with proteomics? Someone here? No? You're familiar. It's complicated, isn't it? Right? Yeah, it's so difficult. I mean, I have no idea how to do that. But there are people, that work with us in the research group. They run the technique and they give you a lot of information. But this is like a screening. It's a screening to know what's going on. But when you do the screening, we observe that people with caries, they have different proteins to people without caries or caries-free, differentially expressed. And so those are potential targets for further research. So we can take now isolated, one of those proteins and study them in depth and see what's going on. Well, for example, you could take some of these proteins, put it in toothpaste, and create something that is powerful because it's neutralizing the activity of acids, for example. That would be a strategy. And if you see this is a heat map, it's called, but this is the same thing. So high caries people tend to group into these hot areas, and low caries risk tends to group into these hot areas. I don't want to go into details. And then we did lipidomics. This is quite novel. Lipidomics is the profile of lipids in your saliva. And when we did that, we also found differences. So we detected 6 new fatty acids identified highly expressed in the high caries group. That means that you can have like a combined profile of proteins that are specific for caries-free people and lipids that are specific for high caries people. And then if you run these experiments then with something similar to how how do you say that, glucometer, the glycemia, to measure the glycemia with the strips, right? You could detect, oh, you have this protein, for example, that you are at high risk. But I'm not saying or arguing, as I said before, that we already have that. But there is room for developing these kind of technologies. These guys are interesting. They're called antimicrobial peptides, are small peptides that are related with health. Beta defensin 1, 2, and 3, LL37, and histatin 5. And all of them were highly expressed in caries-free people, arguing that caries-free people have a better immune system than high caries people. Again, this is what I'm showing. I'm not arguing a more sophisticated relationship with leukocytes or other cells or stuff like that. But what I'm saying here is this is interesting because it shows that at the innate immune level, which is the antimicrobial peptides, people with or without caries lesions don't behave the same. These are the antimicrobial peptides. The thing is, now we know that the nutrients, I already told you, right? The nutrients can protect you against caries, proteins, lipids, and polyphenols. But how long does it take for food to exit the mouth? 2 minutes or 3 minutes? And then it's gone. So we wondered whether that small passing of the food in the mouth was enough to create the benefit or the harm. And we were wondering if there is like a systemic effect of the diet in your oral health. And this is the focus of a new grant that we started, and I want to show you some of the results. So the idea is that when you eat, the food goes rapidly to your gut. From the gut, pass to the blood, and from the blood, it accumulates certain molecules in your salivary glands, and through the secretion of saliva, your saliva is going to be different because you ate this or that. It's not the same. And in turn, that saliva will influence the oral biofilm. And so the biofilm that is all the time in your mouth, because it's not like something that appears 2 hours and then it's gone. It's all day long there. And saliva is being secreted all day long. So that biofilm is going to be influenced by the saliva that you are receiving and by the nutrients that you are consuming 3 or 4 times a day. This is the theory behind. Where did we get that from? Well, there's some evidence, indirect evidence, telling us, for example, that when you eat green leaves, let's say lettuce or spinach, that are rich in nitrate. This is the work of Bob Rosier and Alex Mira from Spain. When you eat nitrate, you have a peak in your mouth that is gone in 2 minutes, and then several hours after, you have another peak in your saliva of nitrate because of the recapturing of the nutrient in the mouth. So using the same principle, we did all the research we did. I mean, we started before, but what I'm trying to say is that there is evidence showing that there is this recapturing of nutrients. So what is the dietary impact on blood and saliva composition and the implication for caries prevention? So this is the experiment. So we recruited, again, 12 dental students that wanted to participate. In this case, it was a much more complicated study because we needed to feed them for 4 days with a Mediterranean diet, 7 days in the phase 1, 4 days with a Mediterranean diet, 7 more days, another washout, and a final 7 days. So we basically converted our lab into a supermarket. Really, there was a lot of food in the lab. And so they had to come to receive the food, and they had to prepare according to, in the project, we recruited nutritionists, dietitians, that were, delivering specific feeding program to the volunteers. And so we took like a baseline blood and saliva sample, then at 4 days saliva, and then at 7 days saliva and blood. And we used something that is called a crossing over design. I don't know if you're familiar with that, but it's, the idea is that 4 of the 12 started with proteins, and then 4 with fatty acid, and 4 with carbs or carbohydrates, and not all of them with the same diet at the same time. And the reason is because there is some bias. So we want to avoid that people eating proteins, for example. I mean, if everyone was eating protein in the beginning, probably they would be much more willing to follow the pattern or the diet. But then by the end of the experiment, they would say, I'm tired. And so that will impact the carbohydrates, not because of the carbohydrates, but because of the motivation of the students. So the way to get around that is to create this crossing over design. So you divide the group into the 3 diets and you mix. All right. So the bias exists, but it's equal for the 3 diets. Is that clear? Yeah. Good. And so we measured all that, the same thing that I showed you before, et cetera, the proteomics, lipidomics. But what I want to show you is the blood analysis. And I think it's important or interesting too. So for the high protein diet, here's meat, but it can be legumes, or it can be eggs or milk or dairy products in general, right? So there was a decrease in uric acid and an increase in uric nitrogen in blood. In saliva, there was an increase in ammonium, and there was a positive correlation with blood, uric nitrogen, and albumin in the saliva. What are the implications for caries of these findings? There's a potential protective effect because elevated ammonium neutralizes acids, potentially reducing dental caries risk. I don't know if you're familiar with the arginine that Colgate put into the toothpaste. Arginine is an amino acid, right? And just like any amino acid is able to buffer the acid. And so it makes sense that when you eat food that is rich in proteins, you have some changes in your blood, and in turn, changes in your saliva that can potentially benefit in caries. And we are very happy with this because this was our theory, and I think this is the first time that someone shows a systemic effect of diet on something that can explain why a high protein diet is beneficial for caries. So I think this is the central message of this one. Then with the high lipid diet, we observe a decrease in transaminases, which is good. It means that the liver is working well. And a decrease in LDH, which is the bad cholesterol, which is also interesting because you would expect. Well, now I have to say, when you put a high lipid diet, this is good fat, it's not saturated fat. I mean, we couldn't do it for ethical reasons, and we don't want to do it either. This is good fat. It's like lots of olive oil, lots of fish, a lot of things that are nuts. But in saliva, we couldn't detect an effect. Although we saw changes in blood, we didn't observe changes in saliva. our conclusion for this in caries is that this is like a neutral effect. However, you have to consider that this diet was imposed for only 7 days. And when we talk with the nutritionists and some people that are, more used to this kind of studies, they said that in order to see changes, probably you need to go to a dietary pattern that lasts for at least 6 months or so. And of course, this type of approach, experimental approach, where you feed people, is impossible for 6 months. I mean, there's no budget that is, enough to support that. So, and yet, with the proteins, we observe changes in saliva and blood. And for carbohydrates, there was an increase in insulin and I don't know how you pronounce it, OMA. OMA is a rate between glycemia and insulin. So when you increase the OMA, that means your sugar metabolism is not under control. So there's a dysregulation in your blood metabolism, sugar, glycemic control. So this is not a good thing. So when you increase carbohydrates, and this increase was not with sugars, with just carbohydrates, all right? There was an increase in these two substances. There was a decrease in pH. You would say, yeah, it's not much, but it's statistically significant, which means that there is more acidic pH in the mouth, so that's a more acid activity. And there was a decrease in buffer capacity. So there was a negative correlation with insulin and OMA. So the impact on caries is that a diet high in carbohydrates may be harmful because acidic saliva environment promotes dental caries, as we all know. So a summary of findings, a high carbohydrate diet creates acidic saliva conditions linked to increased caries risk. A high protein diet elevates beneficial salivary ammonium, potentially protective against dental caries. And a high lipid diet creates limited impact on oral health markers. These results highlight the importance of right dietary choices that support optimal oral health conditions. So basically, as a summary of the idea, the conceptual idea, so when you have dietary intake with proteins or the other macronutrients, there's a gut digestion, there's a gut production of metabolites. This is very important. Nowadays, this is a hot topic in research. The gut microbiota is super important, and we are considering the gut microbiota as an organ that produces molecules that are otherwise impossible to get. So the health of your bacteria in the gut is key. And then there's a blood absorption, a blood circulation that goes to the salivary glands, then a recapturing by the salivary glands, a change in saliva composition, and finally, there's a systemic impact on caries and oral health that may be positive or negative. This is our conclusion. I mean, but you need to understand, this is science. So this is not a clinical guideline or anything. It's just our findings. So the practical recommendations is to reduce carbohydrate intake, particularly sugars, and incorporate protein-rich foods. Balanced diet may benefit with both general and oral health, or health in general, that's better to say. So conclusions, due to its composition, saliva is a potent anti-caries agent. Saliva shows very high variability in its composition that precludes the use of single molecules as caries biomarkers. Caries-free people have a differential biochemical, proteomic, and lipidomic pattern than adults with a high burden of caries. Specific dietary patterns influence blood changes, which in turn determines salivary biochemical composition that may impact on caries. May impact on caries. I'm careful with that. More complex multivariate caries risk assessment systems that include salivary profiles and fingerprinting, validated in controlled clinical studies, need to be developed. So there's room for more research. And with that, I would like to acknowledge the people working in these projects, including Katerina. And this is my email in case you want any of the things that I mentioned. And if there's any question, I'd be more than happy to take it. Thank you.